Hello class, today we are going to learn about respiration. There are 3 stages in cellular respiration. One is glycolysis, second is Krebs cycle, and the third is electron transport chain. Glycolysis is the first stage of cellular respiration and it occurs in the cytoplasm of the cell. In glycolysis, glucose molecules are break down to pyruvate. In here 2 pyruvate molecules are produced from 1 glucose molecule. Now, before we can get to the next stage of cellular respiration, the Krebs cycle, there's some prep work that needs to be done. This is known as link reaction. Each pyruvate, which is produced in the cytoplasm, enters the mitochondria to be converted into acetyl coenzyme A. Remember that 2 pyruvates are created from glycolysis, meaning 2 acetyl coenzyme molecules are produced. This reaction links glycolysis to the citric acid cycle. Now, we are going to talk about Krebs cycle and electron transport chain. The second step of respiration, Krebs cycle, takes place in the mitochondrial matrix.
In Krebs cycle the coenzyme molecule separates, donating the acetyl group to oxaloacetic acid and produce 6-carbon molecule called citric acid. In second step, citrate is converted into isocitrate. This is actually a 2-step process, involving first the removal and then the addition of a water molecule. In the third step, isocitrate is oxidized and releases a molecule of carbon dioxide, leaving behind a 5-carbon molecule, alpha-ketoglutarate.
Step 4. The fourth step is similar to the third. In this case, it's alpha-ketoglutarate that's oxidized, reducing NAD+ to NADH and releasing a molecule of carbon dioxide in the process. The remaining 4-carbon molecule picks up coenzyme A, forming the unstable compound succinyl-CoA. Step 5. In step 5, the CoA of succinyl-CoA is replaced by a phosphate group, which is then transferred to GDP to make GTP. The 4-carbon molecule produced in this step is called succinate. Step 6. In step 6, succinate is oxidized, forming another 4-carbon molecule called fumarate. In this reaction, 2 hydrogen atoms, with their electrons, are transferred to FAD, producing FADH2. Step 7. In step 7, water is added to the 4-carbon molecule fumarate, converting it into another 4-carbon molecule called malate. Step 8. In the last step of the citric acid cycle, oxaloacetate, the starting 4-carbon compound, is regenerated by oxidation of malate. Another molecule of NAD+ is reduced to NADH in the process. In this lesson, we'll learn about the finale of cellular respiration. The electron transport chain uses products from the first 2 acts of glycolysis and the Krebs cycle to complete the chemical reaction that turns into usable cellular energy. Electrons from NADH are oxidized by complex 1 and NADH dehydrogenase. Complex 1 then transfers these electrons to ubiquinone. Complex 2 succinate dehydrogenase. Oxidation of succinate in the citric acid cycle is catalyzed by this complex, and the reducing equivalents are transferred via the FADH2 and a group of iron-sulfur proteins into the ubiquinone pool. Complex 3 cytochrome BC1. This complex oxidizes reduced ubiquinone and transfers the electrons cytochrome C. Cytochrome C oxidase is the terminal oxidase and brings about the 4-electron reduction of O2 to 2 molecules of H2O.
Created using Powtoon.